for time, I think we'll have to move on. So our final presenter before lunch is Carolyn Mockamer. Carolyn is professor of cell biology, and as an expert on coronaviruses, she has been particularly busy these days. Today, she's going to talk with us about her research and what we know about the basic biology of these viruses.
Okay, thank you very much. Need to having a little trouble screen sharing here. Sorry about this.
Is the screen share icon at the bottom of Zoom not working, or?
It looks like I have some privacy thing on I'm using a different computer, which is this stupid. See if I can. Anybody know where the I'm trying to find my privacy settings because it won't let me share the screen.
Are you on a Mac or a PC?
Yep, I'm on a Mac.
So usually you have to allow privacy by going into the, pulling down the Apple menu, going into the system preferences, and then once you're in system preferences, there's a panel that
Yeah, I've got the panel. I don't see the
security. Oh wait, no, that was, never mind. And then, so you're looking at security and privacy.
Yes.
And usually it's under.
Oh, wait, here we go.
Privacy screen.
It's coming up to screen recording, but that's kind of weird.
That's okay.
Okay.
Yeah, we are recording this. That may be why it's.
Okay, there we go. Sorry about this. Luckily, my presentation is pretty short. Okay, here we go.
Perfect.
Sorry about that, everybody. Okay. So I imagine that many of you have, can you hear me okay?
I can hear you, but now we're seeing your notes as opposed to your slideshow. So you'll want to switch. You go up to the top. Go ahead and start the slideshow.
I want to do my presenter view. Okay, well, we'll just,
yeah.
That's fine.
There you
How's
go,
that?
you got it.
Okay, sorry about that, everyone. So I imagine some of you haven't heard the word coronavirus until this January of this year, but I've been fascinated by these viruses for about 35 years now. And I should say at the onset that our work doesn't directly impact precision medicine, which is the theme of this boot camp, but it illustrates, I think, the foundational work that's required to understand a disease. So first, let's just look at the fact that there are many kinds of viruses that infect humans. They can have RNA genomes, they can have DNA genomes. These genomes are wrapped by different types of proteins, and then they can have envelopes surrounding them. And this is like a fatty lipid membrane derived from the host. And what's important here is that these viruses have very streamlined genomes, and that means they depend on the host machinery for their replication and all other things they do to the cells. So many mechanisms for hijacking cells exist by these different viruses, probably at least as many as there are types of viruses. Now, in terms of coronaviruses, the diseases, they infect all vertebrates and generally cause mild respiratory or gastrointestinal disease, including up to 30% of the common cold in humans. But in December of 2002, the severe acute respiratory syndrome coronavirus emerged with 10% mortality, so pretty serious. In 2012, the Middle East respiratory syndrome virus appeared with 35% mortality. And now in January 2020, of course, we have COVID-19 caused by SARS coronavirus 2. And importantly, we don't have any approved vaccines or therapeutics for any of these serious human coronaviruses. Now, these viruses are transmitted to humans zoonotically, which means from an animal. And it looks like bats are the main reservoir for most animal coronaviruses, and they can pass often through an intermediate host before infecting humans, although we don't yet quite know how SARS coronavirus 2 was transmitted to humans. In spite of all the havoc that SARS coronavirus 2 is causing around the world, the structure is quite simple. This cartoon shows a virion with an RNA genome. The RNA is about 30,000 bases long, and it's wrapped by a protein called N, or nucleocapsid. And enclosing that genome is an envelope made of a membrane that has 3 other proteins in it. The M protein is the most abundant in scaffolds assembly. The small protein E is not very abundant in the virus, and I'll tell you more about that in a moment. And then the spike, or S protein, which protrudes quite a distance from the envelope and interacts with the host cell by binding it and allowing entry of the virus. And that S protein also gives the characteristic corona-like structure seen in this electron micrograph that gives the coronavirus family its name. This is the genome, a diagram of the genome of the SARS coronavirus 2. And amazingly, it has just 16 genes that encode for 29 proteins, and they're shown here. Now, just to put that in perspective, your cells have about at least 20,000 genes. So here's this virus causing this huge pandemic that has this very limited amount of genetic information. The first part of the genome codes for proteins that are important for replicating the genome and transcribing the messages. The structural proteins I mentioned are encoded in this part of the genome, along with what we call accessory proteins, which mostly are thought to interact with the innate immune system and try to block that. Now, importantly, envelope viruses, all envelope viruses, use a secretory pathway in the cell. This is a cartoon of the secretory pathway. This is a pathway used by all host proteins that are going to be secreted out of the cell, like antibodies or insulin, for example, or for proteins that are going to be inserted here at the plasma membrane of the cell, such as receptors. And envelope viruses use this pathway for synthesizing their membrane proteins just as well. The cell doesn't know that there's any difference. It's making these proteins because it looks like a normal cell message to be synthesized. So the place where this group of proteins are synthesized is called the endoplasmic reticulum. The next compartment along the pathway is called the Golgi complex. This is has a funny name. It was named after an Italian neuroanatomist who discovered the organelle in the late 1800s. And then these things move in these vesicles to be transported to the cell surface. So you can imagine a virus like influenza making these membrane proteins. The membrane proteins come together, they bind to the genomes, the virus buds out, taking a little piece of the plasma membrane, and now the virus is released and is ready to go on and infect a new cell. But coronaviruses don't do this simpler pathway at all. They have a very unusual way of assembling in the cell. This is just a cartoon showing an overview of the coronavirus infection cycle. That S protein binds a cell receptor that allows entry of the virus. The genome looks like any other messenger RNA to the cell, and it's translated to form those first 2/3 of the genome proteins that are important for replication and transcription. Once these structural protein RNAs are made, the nucleocapsid or N protein is synthesized in the cytoplasm to wrap the new genomes, whereas these membrane proteins go through this pathway I described, the so-called secretory pathway. And they actually do not go all the way to the cell surface. They actually congregate together in this Golgi complex compartment for assembly. And our work over the years has studied these later steps, the assembly and egress of virus from the cells, because this is not the most efficient method for release of virions. So here's just looking at a little more carefully at this. So we have here the 3 envelope proteins, and they're made in the secretory pathway, coming together in this early compartment of the Golgi. And what you see here in this enlargement is that these proteins interact with each other and interact with the nucleocapsid wrapped RNA genome and form a bud. Instead of out of the cell, they form into the inside of this Golgi compartment. And then you have these enveloped virions that have to get through the cell and get out of the cell. And this assembly pathway really presents several challenges for the virus. As you can see here, this is an electron micrograph section through an infected cell. And the Golgi stacks, as I said, they look kind of like stacks of pancakes, and that's why we've been fascinated by the structure function of this organelle. I should also say that Golgi complex, I believe, is the true post office of the cell. Unlike sort of Ken Whitwer's analogy of the extracellular vesicles, that's sort of the postal system, and whereas this is the post office where sorting and processing of cargo goes on. Anyway, the challenge is, first of all, the cargo here, which is the virus, is much bigger than the space between these flattened sets of membranes. So the organelle has to be dilated to accommodate the budding virions. And then the next challenge is how these are going to get out of the cell. Again, it's a slow and inefficient process, probably due to the size of the cargo. So we've been studying this process and wondering, is there a way that we can interfere with it? And over the years, we've studied a protein called the E protein. This is that small envelope protein of the virus. It's only 74 to 124 amino acids, depending on the coronavirus. And we've used a model coronavirus in our work, but we've also studied the original SARS coronavirus. And the first function for this little E protein is in assembly, where the E protein either helps bend the membrane to form the bud and or helps it undergo scission from the Golgi membrane to allow these virus particles to be inside the Golgi itself. But we've also found another interesting role for the protein, and that is in modification of this secretory system. And this role requires the E protein that's in the membrane itself and not incorporated into the virus. Remember, I said this is a very low abundance protein in the virus particle itself, but it's present in pretty high levels in the cells, and it sits in the Golgi membranes, and through its activity modulates the environment of this compartment, and that allows these virus particles to exit the cell in an infectious form. We know that because a mutant of this E protein that can't do this function, the particles that are released are damaged, and they've run into enzymes that chew up the proteins, and therefore they're not infectious. So this small little protein has 2 functions, and the 2 different functions depend on different portions of the protein. And I think this is a really nice example of how these streamlined genomes of the virus can perform the multitude of functions that they need to. But the big question here, and I hope you're thinking about this, is why in the world would a virus want to do this? What is the advantage of intracellular assembly with all these challenges that the virus has? And I wish I could say that we knew the answer to this. We don't know the answer. We have ideas that we've tested over the years, but clearly it provides a real advantage for the virus, given that these viruses can mutate and do it better. So that's a question that we're really very still interested in, and we just don't know enough about the basic biology of these viruses to be able to answer the question yet. Now, importantly, the fact that the vaccine, there's not going to be a vaccine in the near future, and so what about therapeutic approaches? Well, there's drugs that block the entry phase. There's drugs that interfere with replication of the virus, but nothing yet that we know of that interferes with assembly or release. And part of the problem here is because this part of the pathway hasn't been well studied. And if there's one good thing to come out of the COVID-19 pandemic, I think it's that many, many cell biologists have become very interested in these viruses and now want to study this process, this assembly and release process. And hopefully with sustained funding, we'll be able to get another approach to interfering with coronaviruses, because clearly pandemics in the future are certainly not impossible to imagine. And I'll just close with thanking a number of trainees, students, postdocs, undergraduates who have been involved in the various aspects of coronavirus biology, including all of these graduate students and postdocs here who were involved in the latter part of our work and our funding sources. Thank you.
Okay, great. Thank you very much, Caroline, for that terrific overview. There aren't any questions that have come in just yet, and we're running over time. So I think what we're going to do now is just go ahead and break for lunch.